Greetings to one and all present here. I'm Niharika Pandala, and my research topic is a preliminary investigation in the molecular basis of host shutoff mechanism in SARS coronavirus. Severe acute respiratory syndrome coronavirus, also known as SARS coronavirus 1, is a serious form of pneumonia. It causes difficulty in breathing and, in severe cases, leads to death. SARS coronavirus 1 contains a single stranded RNA genome that replicates in the cytoplasm of the host cells. The replicated gene contains two-thirds of the genome that encodes for the two larger precursor polyproteins cleaved into 16 non-structural proteins, NSPs, out of which NSP1 is the first to be translated. NSP1 is known to suppress host immune response and also acts as host shutoff factor by dampening host gene expression. This protein is a 180 residue long protein that consists of two domains. Residues 1 to 114 is a well-defined structure studied by NMR spectroscopy, as shown to the image to our left. Residue 115 to 180 is poorly characterized due to its dynamical properties, which makes it difficult to be studied by traditional methods. The methods used to investigate the host shutoff mechanism of non-structural protein 1 included the following steps. Step 1 was computational protein modeling of 26 mutations of NSP1 that demonstrated both amplifying and attenuating activities of its host shutdown property in laboratory settings. The model was achieved using ITASR, which stands for Iterative Threading Assembly Refinement. The second step included VMD and NAMD software suite to investigate the structure-function relationship of NSP1. Here, VMD stands for Visual Molecular Dynamics and NAMD stands for Nanoscale Molecular Dynamics. Within this method, effect of charge distribution, protein energetics using NAMD energy, and topological variation considering volumetric occupancy were explored. The third method considered binding site recognition using MS-TALLY software, where MS-TALLY stands for Multiple Structure Torsion Angle Alignment Technique. This software is used for identifying an active site based on evolutionary relationship between a group of proteins with known similar substrate binding properties. The final and the fourth method was mining of protein data bank with PDB-Mine software. PDB-Mine was utilized to discover any existing protein structure with a similar k-mer sequence to NSP1 protein. K-mer is the number assigned by the user to find a rolling window size of the given number to query the protein data bank. On analyzing the laboratory-based mutation of NSP1, we mainly explored two potential regions of interest, as shown in the table here. Both of them inhibit host gene expression by amplifying NSP1's function. Mutation 1 is E55R, E57R, K58E, G59R, while mutation 2 is R73E, D75R, L77A, S78E, N80G. Here, all the alphabets represent amino acids, while the numbers represent the location within the 180 residue long protein. To understand this better, let me give two examples. Considering mutation 1's first point mutation, which is E55R, stands for E glutamic acid, at location 55 is substituted by R, which is arginine. Similarly, mutation 2's first point mutation, R73E, can be understood as R arginine. At location 73 is being substituted by E, which is glutamic acid. Now comparing and contrasting both these mutations with respect to the features we studied, The first being effect of charge distribution, which would take into consideration amino acid substitutions. We can see the images on the top as surface representations that show acidic amino acids in red, alkaline amino acids in blue, and neutral amino acids in yellow. So mutation 1 appears as an outward protrusion, and we see it exposes positively charged alkaline amino acids on the surface. This may facilitate the interaction with negatively charged backbone of RNA. While mutation 2, in contrast, appears around a loop and changes the amino acid residues to primarily acidic residues. On considering protein energetics, which is measured in kilocalorie per mole, we see that the wild-type NSP1 has a protein energy of negative 12,000, which when compared to mutation 1, which is at negative 16,000, stabilizes the protein, while mutation 2, which is at negative 10,000, destabilizes the protein. Considering topological variation, which considers volumetric occupancy, we see that the substitutions form hydrophilic nodes. Arginine substitution here contributes to long-range connectivity and positive charge. While mutation 2, we see most mutations are hydrophilic. They have a relatively small difference in side chain length compared to the wild type. The rationale we arrive for mutation 1 is that mutation 1 facilitates cooperative stabilization of native conformation, altering binding sites' binding properties, while mutation 2 has the potential for a highly cooperative transition to interact by short-range interactions. The MS tally conservation score for NSP1 protein with respect to other similar proteins yields the following graph. We see 4 peaks, out of which mutation 2 lies in the region of 74 to 80 that has the highest peak with complete surface exposure compared to other 3 peaks which have partial surface exposure. It can be hypothesized that the 3 critical and buried conserved regions are the requisite structural requirement to form the structure of this protein that facilitates the docking process at residue 74 to 83. PDB-MIND then was used to discover existing protein structures with a similar chimer sequence to NSP1 protein. We see that sequence FIKRSDA at region 70 to 76 has a lot of proteins that match the particular template. And then we have the other 2 bars. So basically, we created 174 6-mers from a rolling window size of 6 and 7 residues, which showed the most significance in structural inference. And these inferences can be used to construct the protein's fragment structure or assist in discovering their binding sites. Our conclusions based on structure-function relation by investigating energetics is that mutation 1 stabilizes the protein, while mutation 2 destabilizes the protein. Both the mutations function by distinctly different mechanisms. Based on charge and surface perturbation, we can say mutation 1 has positively charged alkaline amino acids due to the presence of arginine, while mutation 2 does not change charge distribution significantly. Yet, both the mutations amplify the host shutoff function of NSP1. On investigating the side chain interactions in the mutated NSP1, we can say mutation 1 may favor long-range binding and interaction with nucleic acids, while mutation 2 might support binding host factors due to its inherent flexibility. Flexibility is the key feature of NSP1. Each mutation can function differently in the presence of a host factor, which is a RNA or a protein. PDB-MIND and MS tally studies show no much significant results for the first mutation, but for the second mutation, it's being identified for ribosomal binding function, and the surface exposed conserved region supports this region's binding propensity. Critical binding sites and overlapped results provided by both the softwares provide information for future investigation.
Thank you.